Proliferation is an important measure of cell function. This protocol allows investigators to develop a sensitive proliferation assay in their labs without the high cost of commercially available kits. The main advantage of this technique is that it avoids harsh treatment of cells and/or the use of radioactivity. Demonstrating this technique will be Vicki Wong, the manager of my laboratory.
To assay proliferation, remove vascular smooth muscle cells from a flask with smooth muscle cell media in 2% fetal bovine serum and plate at 20,000 cells per milliliter in DMEM and a 96 well plate. Grow the cells at 37 degrees Celsius overnight. Add 30 nanograms per milliliter platelet-derived growth factor to 3 to 6 wells as a positive control to stimulate proliferation. Then, add PBS to the same number of wells as a negative control. Incubate for 72 hours. Dilute previously prepared 5 millimolar EDU stock to 1 millimolar and add 2 microliters to each well for the last 24 hours of the 72 hours culture period. Keep one set of replicates without EDU to determine background fluorescence and luminescence. 24 hours later, fix the cells by removing media, then adding 150 microliters per well of 4% paraformaldehyde, and incubate 10 minutes at room temperature. Remove the paraformaldehyde and add 150 microliters of 1% TX100 in water per well and incubate at room temperature for 30 minutes. Wash 3 times with PBS to remove the TX100. To detect incorporated EDU using fluorescence, make 10 milliliters of labeling.